हाय एवरीवन, वेलकम बैक टू माय यूट्यूब चैनल मैथ्स इजी वे। आज हम स्टैंडर्ड 9 का साइंस चैप्टर नंबर 16 हेरिडिटी एंड वेरिएशन के एक्सरसाइज सॉल्व करेंगे। एक्सरसाइज है फर्स्ट है फिलअप कंप्लीट द फॉलोइंग सेंटेंसेस बाय चूजिंग द अप्रोप्रिएट वर्ड्स फ्रॉम द ब्रैकेट। ब्रैकेट में कुछ वर्ड्स भी है, उसे हमें फिल करने का है। तो फर्स्ट है hereditary character are transferred from parents to offspring by dash. Hence they are said to be structural and functional unit of heredity. तो यहाँ पर क्या आएगा by genes, यहाँ पर दिया हुआ है। नेक्स्ट देखते हैं organism produced by dash shows minor variation. इधर आएगा asexual reproduction. Sexual reproduction. Next देखते हैं the component which is in the nuclei of cells and carries the hereditary characteristics is called dash is chromosome. Okay, next देखते हैं chromosome are mainly made up of DNA. Next है organism produced through dash shows major variation. इधर आएगा sexual reproduction. Sexual reproduction. Next question आंसर देखते हैं। क्वेश्चन नंबर 2 है explain the following. A. Explain Mendel's monohybrid progeny with the help of any one cross. Answer. Mendel's monohybrid cross is a cross between two parents that has one pair of contrasting characters. Monohybrid means one pair of contrasting character है. Mono means one pair. Okay. For example, if pear plant with yellow seed coat is crossed with pear plant having green seed coat, then in the F1 generation all the plants produce yellow seed. Okay, उसका any one cross लिखिए, ये लिख लीजिए। नेक्स्ट देखते हैं एक्सप्लेन Mendel's D-hybrid ratio with the help of any one cross. Answer. Point 1. In D-hybrid cross, Mendel considered two pair of contrasting characteristics. D-hybrid मतलब D मतलब two pair of contrasting characteristics. Second point, he crossed pear plant having round yellow seed with pear plant having wrinkled green seed. Thus, for dihybrid cross, he considered two characteristics, that is seed color and seed shape. Third point, Mendel's experimental of dihybrid cross. तो ये डायग्राम है, चार्ट है, सॉरी डायग्राम में चार्ट है, आप ये देख लीजिए, ये कॉपी कर लीजिए। नेक्स्ट देखते हैं। Next is see difference between monohybrid and dihybrid cross. Answer है monohybrid cross and dihybrid cross. Monohybrid cross. Point 1, cross between two plants with only one pair of contrasting character is called monohybrid cross. Dihybrid cross, cross between two plants with two pairs of contrasting character is called dihybrid cross. पॉइंट सेकंड देख लेते हैं। F1 hybrid of monohybrid cross produce two types of gametes. Dihybrid cross में F1 hybrid of dihybrid cross produce four types of gametes. थर्ड पॉइंट है monohybrid cross का gametes produced by F1 hybrid are similar to gametes produced by the P1 generation. डायहाइब्रिड क्रॉस का है only 2 gametes, but of only 2 gametes out of 4 produced by F1 hybrids are similar to those of the P1 generation. फोर्थ पॉइंट देखिए, मोनोहाइब्रिड क्रॉस का फिनोटाइप रेशियो इन F1 generation of monohybrid cross is 3:1 and dihybrid का है phenotypic ratio in F2 generation of dihybrid cross is 9:3:3:3:1. ओके, ये देख लीजिए, ये आप लिख लीजिए। 2 मार्क्स का रहेगा तो 4 पॉइंट लिखना पड़ता है। चलिए नेक्स्ट देखते हैं। क्वेश्चन नंबर डी है, इज इट राइट टू अवॉइड लिविंग विथ ए पर्सन सफरिंग फ्रॉम ए जेनेटिक डिसऑर्डर? आंसर नो, इट इज नॉट राइट टू अवॉइड लिविंग विथ ए पर्सन सफरिंग फ्रॉम ए जेनेटिक डिसऑर्डर। सिर्फ नो लिखकर नहीं छोड़ सकते, कभी 2 मार्क्स का रहेगा तो हमें कुछ एक्सप्लेन भी करना रहेगा। सो देखिए पॉइंट्स, हैज दे आर इनहेरिटेड फ्रॉम पेरेंट टू ऑफस्प्रिंग only and they are non-continuous, thus they do not spread through contact. Third point है, also people suffering from an incurable genetic disorder may require help and support and should not be avoided. नेक्स्ट देख लीजिए, एक्सप्लेन। क्वेश्चन नंबर 3 है, आंसर द फॉलोइंग क्वेश्चन इन योर ओन वर्ड्स। ये है, व्हाट इज मीन बाय क्रोमोसोम? एक्सप्लेन इज टाइप। आंसर पॉइंट नंबर 1, क्रोमोसोम इज द स्ट्रक्चर इन द न्यूक्लियस ऑफ सेल्स दैट कैरीज द हेरिडिटरी कैरेक्टरिस्टिक। पॉइंट नंबर 2, दे आर मेनली मेड अप ऑफ न्यूक्लिक एसिड एंड प्रोटीन्स। पॉइंट नंबर 3, depending upon the position of centromere and the length of the chromosome arms, there are 4 types of chromosome. ये 4 टाइप के क्रोमोसोम है, मेटासेंट्रिक, सब मेटासेंट्रिक, एक्रोसेंट्रिक एंड टेलोसेंट्रिक। डायग्राम बहुत इजी है, इसका एक्सप्लेनेशन भी बहुत इजी है, जैसे शेप बना है उसी हिसाब से हमें बताने का। ये जो पॉइंट है इसको सेंट्रोमेर बोलते हैं। ओके, फर्स्ट टाइप है मेटोसेंट्रिक। सेंट्रोमेर इज इन द सेंटर और एट मिड पॉइंट ऑफ द क्रोमोसोम। बोथ द आर्म्स ऑफ क्रोमोसोम आर इक्वल इन लेंथ। क्रोमोसोम अपीयर हैज इंग्लिश अल्फाबेट वीके से बना है। सेकेंड है सब मेट्रोसेंट्रिक। सेंट्रोमेर इज समवेयर नियर द मिड पॉइंट ऑफ क्रोमोसोम। वन आर्म इज स्लाइटली शॉर्टर देन अदर। क्रोमोसोम अपीयर हैज इंग्लिश अल्फाबेट एल, जैसे दिखता है वैसे हमें लिखने का है। थर्ड पॉइंट है एक्रोसेंट्रिक। द सेंट्रोमेर इज नियर वन एंड ऑफ द क्रोमोसोम। वन arm is much smaller than other. Chromosome looks like English alphabet J. Third is telocentric. The chromosome is right at the one end of the chromosome. It has only one arm and appears as English alphabet R. Question number B है Describe the structure of the DNA molecule. पहले मैं आपको स्ट्रक्चर डायग्राम दिखा देती हूँ। डायग्राम आपके टेक्स्ट बुक में दिया हुआ है, यही डायग्राम आपको पहले बनाने का है। पॉइंट 1 है In 1953, Wilson and Crick proposed a model of the DNA molecule. Second point, according to this model, the two strands of nucleotides coil around each other to form double helix. Third point, this double helical structure appears as flexible leather. Fourth point, each strand is made of nucleotides. These nucleotides are the unit of DNA. 6 point. A molecule of a nitrogenous base and phosphoric acid are joined to a molecule of sugar to form nucleotide. 6 point. There are 4 types of nitrogenous bases in the form of adenine and urine. Purines को कहते हैं thymines and cytosine, pyrimidines. Thus 4 types of nucleotide are formed. 7 point है the nitrogenous bases from the 2 strands joined by hydrogen bonds to form base pair, rungs of ladders. 2 threads form long chain of alternative joined molecule of sugar and phosphoric acid, rails of ladder type. 8 point है the adenine always pair up with thymines and urines always pair up with cytosine in the DNA molecule. Next picture. Express your opinion about the use of DNA fingerprinting. Answer point number 1. DNA fingerprinting is a technique used to identify the sequence of genes in the DNA of a person, that is, genome of the person. Second point है this technique helps in identifying the linkage of the person. It is also used in identifying criminals, resolving paternity and maternity dispute, etc. Third point is it also involves lot of complexity as it requires testing of more than one sample for accuracy. The information gathered from the DNA test should be kept private and confidential as it can be misused if leaked. Fourth point is the introduction of DNA fingerprinting has helped to resolve several issues of the modern world. Care has to be taken in its implementation and use. Next page. Next is explain the structure, function and types of RNA. Structure of RNA. First point is the nucleotide is the smallest unit of the chain of the RNA molecule. Second, RNA nucleotide is made up of ribose sugar, phosphate molecule and any one of the 4 types of nitrogenous bases: adenine, guanine, cytosine and uracil. Third point is large number of nucleotides are bonded together to form the macromolecule of RNA. Function: RNA along with DNA and ribosomes helps in protein synthesis. Third, depending upon the function, RNA are classified into 3 types. के डायग्राम दिखा देती हूँ. ये डायग्राम है टाइप्स ऑफ आरएनए पे आपके टेक्स्ट बुक में दिया है, आप देख लीजिएगा. डायग्राम देखकर लिखना आसान हो जाता है. फर्स्ट टाइप है मैसेंजर आरएनए, शॉर्ट फॉर्म में लिखा है एम, मैसेंजर का एम, आरएनए स्मॉल लेटर में एम, आरएनए. It carries the information for protein synthesis from genes, that is DNA segments in the nucleus of cell, to the ribosome, that is in cytoplasm of cell. Second है ribosomal RNA, small letter दिया R, RNA. It is the component of cellular organelle ribosome. Ribosome perform the function of protein synthesis. Third है transfer RNA, transfer का T, small letter RNA. It carries amino acid up to the ribosome according to the message on the mRNA. Next. Question number E है why it is necessary for people to have their blood examined before marriage. Answer for first point, Due to blood examination before marriage, the prospective marriage partner will previously know about the possible genetic disease that their children might inherit. Using this knowledge, they may choose not to have children or not to get married in the first place, or would be able to take necessary measures at the right time. Second point, on the basis of their past medical histories and the differences in their blood types, it becomes possible to give valuable medical advice to the prospective marriage partners. Third point is blood examination before marriage protects people from contracting contagious disease from the prospective partner. Fourth point, also information about RH capability can be obtained and the couple can be advised about the risks during childbirth if any. Therefore, it is necessary for people to have their blood examined before marriage. आगे का नेक्स्ट वीडियो में सॉल्व करेंगे. टिल देन बाय, टेक केयर.